Hello everybody, my name is Rebecca Carvallar and today I'm going to talk about population modification of mosquitoes for malaria control in Africa. There are two genetic control strategies for mosquito populations. The first one is what we call population suppression. This strategy reduces or eliminates mosquito population. In contrast, population modification is based on changing the ability of mosquitoes to transmit pathogens. This concept was first introduced by Francis Cortes in 1968. We do population modification by engineering mosquitoes with genes. We call those genes effector genes to prevent malaria parasite transmission. We believe that the introduction into a mosquito population of those effector genes will confer resistance to the malaria parasite and should result in less disease and less death. In 2015, it was published the first gene-drive system for mosquito control. It was a population modification system in the Indian malaria vector Anopheles stephensi. The system has two main components, a gene-drive component and an effector component. Among the gene-drive elements, it is included the expression of the Cas9 protein driven by the Basa promoter, which is a germline promoter expressed in the ovaries and testes of the mosquitoes. On the other hand, the effector part of the system is composed by two effector genes, 2A10 that blocks the sporozoites and M1C3 which blocks the okinates. Although the system was really efficient in driving effector genes into a mosquito population, with an impressive 99.5% of drive, it presented some challenges. Among them, the first that we were able to identify, it is the fitness cost associated to the KH locus. In this system, the KH locus was targeted to insert the gene drive system. As a result, the females or the mosquitoes will have a white eye phenotype. And we observed that homozygous female with white eyes die after the blood feeding. The second challenge that we identified is resistant alleles. The resistant alleles limit the ability of the drive to spread into populations. We believe that the resistant alleles are because the highly activity of the Basa promoter, which results in Cas9 maternal effect. In this system, we observe around 5% of resistant alleles or non-homologous enjoining events, which give us no drive in further generations. So far, there are 3 gene drives in mosquito. The first one was the one published in 2015. It is a population modification system for Anopheles stephensi. In contrast, the other 2 systems that are present today are based on population suppression strategies for the Anopheles gambiae African malaria vector. We recently developed a population modification gene drive, and in order to overcome some of the challenges that we observe in the prototype strain, in the Anopheles stephensi prototype strain, we made some modifications. First of all, to overcome the fitness effect of the KH locus, we changed the target site to the cardinal gene. The cardinal gene is as well part of the Omicron pathway. Moreover, we also decided to change the Basa promoter, and instead we placed the Nanos promoter. We believe that the Nanos promoter will give us a more localized expression in the germline cells, and as a result, the resistant alleles will be less. The first question is if we are able to see a phenotype associated to the cardinal target gene. Yes, there is a phenotype associated. The larvae show red eye. The pupa present a gradient coloration in the eye, and the adults also have red eye. And sorry, I'm apologizing for the mistake. The adult pictures are mislabeled. The second question is, does it drive? Yes, it drives. and it drives incredible good. It can reach 100% drive. I think this is the first time that a gene drive system in mosquito can reach 100% drive. And finally, the third question that you might be asking is if we see resistant alleles. No, we don't see resistant alleles. All the mosquitoes, the negative mosquitoes that we recover from each generation, they were sequenced. We analyzed those sequences, and we were able to determine that they are true wild types. It is really nice to tell you that we have a next generation gene drive system in Anopheles gambiae that give us 100% drive with no resistant alleles and no fitness cost. How good does this product have to be? We believe that it must meet the efficacy and safety criteria as well as the regulatory and social criteria, and that's why we recently developed a target product profile for mosquito population modification gene drives, and this is no longer an academic exercise. The most important part is that target product profiles describes the ideal characteristics versus the minimal essential characteristics that the system has to have, and we believe that all this information has to be informed by modeling. Lastly, what it will take to make it happen. In other words, what do we need to do in order to put these mosquitoes in the field? We believe that our best strategy is to start with small, less complex trials, like for example, islands. So we believe that we need to choose a less complex locality to start with our field trials. For example, Santo Tomé and Principe Islands and the Comoros Island in Africa. Those places are very good candidates for a first trial because they are ecologically contained, and the complexity is lower when we compare it with a continental complexity, for example. The second step will be to do field trials in more complex or medium complex localities like bigger islands, to finally be able to extrapolate all that information and knowledge and get to the point that we can go and do a continental release or trial. How are we going to do this? We think that it's going to take 3 phases at least. We are currently in phase 1, which is the laboratory phase. We are developing, we are refining our G-Dry systems. We are testing those systems in cage experiments in the insectary, and this will give us a lot of information to model or refine all the systems that we have in the laboratory. The second step will be the confined open trials, which mean those small islands that I talked in the previous slide. And we are currently engaged with Santo Tomé and Principe and also the Union of the Comoros. We are in conversations with them to try to conduct some field trials there. And I'm very happy to tell you that our proposal for 1 year our proposal for 1 year project has recently accepted, so we will be able soon to start working in those sites to do field site selections, to also do the field site characterization, and of course to do those confined field trials. Finally, we believe the last step will be an open field trial, which will involve a continental, the characterization and selection of a continental field site that can be promising for us to test these gene drive strains that we have. It is really important to mention that during all this process, we are carrying regulatory, community engagement, and communications activities with the authorities of these places, as well as with the community. And about the community engagement, we are currently in contact with local community members and leaders. We are talking with them about our proposal and all the working plans that we have in those localities. We share information with them about who we are, what are we doing, why we are doing, and how we are going to do it. We also provide them with basic education and information about malaria biology. And we also try to answer all their questions and concerns about our activities. And finally, we are trying to understand all the regulatory structures and processes that need to be carried in order that we can release mosquitoes in these places. I just want to say thank you to all of you for this opportunity, and also I would like to thank to all our UCIMI members. We have collaboration with the University of California, Davis, Berkeley, Los Angeles, and San Diego. And of course, thank you to our funders, the UCI Malaria Initiative and the Bill and Melinda Gates Foundation.